better idea of the format too. I'm gonna write that down to think of it, but at least 40 to 50 multiple choice for sure. It's just a question of if I'm gonna have you guys do short answer essay type questions at all. So let me see, format. Okay, those are the announcements. Questions before, any other questions before we continue biological molecules and continue talking about carbohydrates? I think that's where we left off. No, okay, then let's dive in. Oh, well, if we look at this, so we talked about carbohydrates, right, on Thursday. And I believe we went up to a disaccharide, polysaccharide, ringing any bells, monomers, polymers. Did we cover those? So a monomer was that repeating molecule unit. So in a carbohydrate, that was called what again? What was the monomer for a carbohydrate? Monomer for carbohydrate. Monosaccharide. And glucose is an example of a monosaccharide. Another example of a monosaccharide is fructose. No, oh wait, yeah, fructose. Fructose, glucose, these are monosaccharides, and they look like this. There are these 6-carbon ring structures. And yes, I mentioned glucose was a very important one that we'll talk a lot about this semester. This was a disaccharide, which is no longer a monomer, but falls into our polymer category. Polymers are these larger molecules that consist of 2 or more or many monomers that are bonded together to one another by these covalent bonds. What was an example of a disaccharide? Hopefully I mentioned one. Did I mention one? There's a very famous one. Some people are intolerant to it. Lucky enough, I'm not. I love ice cream. Lactose, yes. Although maybe it's a bad thing because I eat too much ice cream. And pizza. Yeah. So lactose, disaccharide, totally. And we're going to look at lactose again today, actually, because we're going to learn a couple new terms and we'll use lactose as an example. And we're also going to look at these polysaccharides. So it's kind of like you have monosaccharide, which is 1 unit, disaccharides, which are 2 units, couple lactose, maltose is 1. Sucrose, table sugar, actually is another example, which is a glucose and a fructose actually linked together. So that's another disaccharide. But after that, we're really talking about hundreds and thousands of monosaccharides strung together in these polysaccharides. And we eat those. So we're going to talk about that as well. Okay, cool. And again, these monomers and polymers, right? We're going to have a monomer for each of the biological molecule classes, of which there are 4 classes of biological molecules. And we're going to have a polymer for each one, except lipids. So 3. Lipids are unique, and if we have time, we'll get to them today. We're kind of ahead of schedule, so we might get to them today. Lipids, they're unique in the sense that they don't have a true monomer, and there's different kind of examples of lipids, and they look completely different. But the 3 other categories that we'll talk about follow this rule of you have a monomer unit that's a very specific looking smaller molecule. And then the polymer version is just going to be a repeating sort of string of these molecules. And our other classes, carbohydrates, proteins, and nucleic acids, follow that rule. But we're going to talk about that. So let's begin by talking about something that is very important. And it's a type of chemical reaction, actually. It's called a dehydration synthesis reaction. All right. A dehydration synthesis reaction. I'm going to talk about why this relates to biological molecules. Okay. It's a big, fancy, long name, but it's not that fancy. Oftentimes in biology, the term, the concept we're learning, the name for it, there's a very specific reason behind why we call it that. And it may seem all scary at first, but you pick it apart and it's like, oh, okay, that makes sense. So let's write dehydration synthesis. This is a chemical reaction that joins monomers together to form polymers. Chemical reaction that joins monomers to build polymers. Very important reaction. Okay, so let's pick it apart. The name dehydration synthesis. First, let's look at dehydration, or actually, let's look at synthesis first. So, synthesis means, what does it mean to synthesize something? Create or build, right? So let's write to build. It's kind of like if you synthesize a new drug, you created it, you built it. I use this word a lot, synthesis. Biologists use it a lot. Okay. What does dehydration mean? What does it mean to dehydrate? Water loss, totally. When you're dehydrated, water has been removed from you, and hopefully you hydrate soon and don't pass out or anything. So dehydration is the loss of a water molecule. Loss of a water molecule. Okay, so far we've got a chemical reaction that's going to build a larger molecule from smaller units, monomers. We're building polymers, so we're building something bigger from smaller pieces, right? Synthesis, we're building, right? And we're gonna lose a water molecule to do that. That's all dehydration synthesis means. Synthesizing a large polymer or a larger polymer by combining monomers with the loss of water. So what does this look like? Let's take a look. What I'm going to do actually is, for my example here, since we just talked about carbohydrates, I'm going to build a disaccharide, lactose specifically. So 2 monomer units, 2 monosaccharides. I'm going to link them together. I'm going to join them together, specifically glucose and another monosaccharide called galactose. Kind of has a cool name. Kind of like galaxy, but galactose. Lactose with gal in front. So let's first draw glucose. Has that hexagon ring structure, right? So this is a glucose. And to form lactose, the disaccharide, lactose is made up of glucose linked up with a galactose. So I'm going to join together a galactose, which is another monosaccharide. In fact, I'm going to write monosaccharides underneath. Monosaccharides. Okay. Now, importantly, on glucose or galactose, it actually doesn't matter. Oh, what is the one on the right says galactose. This is galactose. And this is glucose. Okay, so on one of these molecules, there's going to be a group sticking off of it with an OH, covalently bound to the rest of the molecule. It's this OH group. We call it a hydroxyl group. Okay. And then on the other molecule, there's an H sticking off, just the hydrogen covalently bonded to the rest of it. I drew them a little too close together, but they're sticking off each molecule, okay? There's a hydroxyl sticking off one and a hydrogen sticking off of the other. So what's going to happen, I'm going to draw a reaction arrow. It's a chemical reaction. You have a reaction arrow. And I'm going to combine them and link them together to form lactose. So I'm going to join them right now and form lactose. So it'll look like this. So now both of these units are bound together. This is a disaccharide, right? And it's lactose. So if I write glucose here, glu for short, galactose, gal. But together, when they're bound together, it's a disaccharide called galactose, or I'm sorry, lactose. Lactose, like in milk. Okay, totally ran out of room. Should have shifted it over. Okay, I know what I'll do. So you probably have room underneath in your paper. So I'm just gonna erase this, but just keep it here. It's a disaccharide, it's lactose. And I wanna show you what happens when we combine our monomers here. And this is for any dehydration synthesis reaction. In order to combine our two monomers, our two groups here, our hydroxyl OH and our hydrogen, leave. They come off the molecule. And these two molecules are actually bound together now. And if you see what left, OH and H, that's H2O. Two H's and an O, right? So I'm just gonna erase this, but because the water leaves when the two monomers here join together, we also have to include on our other side of the reaction here, our product side, that we have plus water. That in joining these monomers together, we also get water as a byproduct. Water has left these two molecules when they join together. And there you go, that's a dehydration synthesis. We dehydrated it, we synthesized a larger molecule out of it. Okay. So on the topic of larger molecules, let's go ahead and talk about polysaccharides now. We've talked about a few disaccharides, monosaccharides, but we're gonna step back here and talk about carbohydrates still, and talk about the polymer or other polymers, polysaccharides, the longer chains of monomers strung together, and a few really important examples when it comes to carbohydrates, polymer or polysaccharides. Okay. Okay. This. Okay. Polysaccharides. So you can already see here that these are much larger, right? Like from a few hundred to a few thousand monosaccharides strung together by covalent bonds. And there are three very, very important polysaccharides of this kind that I want to talk to you about. And The first one is starch, here on the top right. And starch is a chain of glucose molecules bound together. So let's go back to the board here. I'm gonna draw starch in green because one of the hallmarks of starch molecules is that they're only found in plants. We don't have starch in our bodies. We eat plants, we eat starch, but we don't have it in animals, okay? So starch is found only in plants. How should I do this? Oh, okay, I know. Before I talk about the function of starch, I'm actually going to talk about another example of a polysaccharide first, because it has the same function. And then I'm going to group them together. So the next example, oh, by the way, starch, you find starch in potatoes and wheat, grains like wheat, even corn. So within the cells of potatoes and wheat and corn, within those cells, you'll find these granules filled with these long carbohydrate chains. And why? Why is that? Well, we're going to talk about it in one second. Before that, I want to talk about glycogen for a second. Glycogen, another polysaccharide example. As you can see, long chain of glucose. Just the bonding is a little different, so it has a tiny different structure. I'm going to write glycogen in red here. You don't find glycogen in plants. You find it in animals. You find it in us. So found only in animals. And actually, fun fact, you'll find glycogen stored within muscle cells and within the liver and the liver cells. So liver and muscle stores an abundant amount of glycogen, which is this branched off, cool looking polysaccharide. Again, a bunch of glucoses bound together covalently. But the important function for starch and glycogen is the same. Found in different organisms, but same function. Storage of energy. I'll just do black. All right. So I'm actually going to go ahead and bracket these guys and write function, energy storage. Storage. So carbohydrates are actually are one of the primary sources of energy that we use, specifically glucose. And when we talk about respiration, you'll see that glucose is this main player in getting energy from food. And basically, without going too in depth here, if we are in a starvation state maybe, or we haven't eaten in a while, our body and plants, especially during hibernation and winter, will start to break down these long chains of stored energy, in a sense, and into the individual monomers, the individual glucose molecules, and use it because we haven't eaten any sugar in a while. So we'll start to break down the glycogen in our liver and our muscles. It takes about 24 hours without eating to actually fully deplete glycogen, interestingly enough. Then you move on to fat, but that's metabolism and another topic. But just being aware that this is their function, energy storage. Okay. There's one more very important polysaccharide that I want to discuss. It's called cellulose. And I'm going to do it in green as well because you don't find cellulose in us, in animals. You find it only in plants. So I'm actually going to divide it too. So it has a different function. Cellulose. Okay. So if you look here, it's just kind of interesting. I like to show cellulose and how it's more of this linear, fibrous polysaccharide that stacks on top of each other. It actually, I don't know if I mentioned, but in biology, oftentimes, where this major overarching theme is always present, where structure, the way the molecules look, or the protein, whatever we're talking about, or the cell even, the cell type, whatever it is, the structure always relates to its function. And here is a good example of that. Cellulose, its function is as a structural support in plants. And if you ever seen a house being built or anything with a firm structure and you know, just something built for protection and for strength, you're going to see this type of layered cable-like structure, right? And that's what cellulose looks like. Just stacks on top of each other in these fibrils. Creates a very tough network, polysaccharide. Yeah. So I'm going to write found only in plants, but more specifically, found only in plant cell walls. When we talk about cells and plant cells, we'll all mention this again, but essentially animal cells, cells of us and other animals, don't have a structure called a cell wall. And it's actually why if you feel your skin or muscle, all our cells, they're flimsy. They're not very strong. They don't have as much, they don't have as much rigidity to the cells. But if you look at plants and trees, they're very strong. They can withstand weathering force, right? Wind and all these things. And that's because their cells have these cell walls. And plant cells actually, they kind of look more rectangular, like bricks. And it allows them to stack on top of each other to form this, strong thing, strong structure. And it's because of the cellulose in the cell walls. So let's write the function for cellulose as support, structural support. Even you could say protection. You know, animals that run by and trample over it, all these things, the cell wall provides that barrier, right? And it's why when you crunch into celery or lettuce or carrots, it's a crunch, right? It's all the cellulose and it's fibrous. It's really good for you, actually. Okay, that's enough of cellulose. I've talked a lot about it. All right. Any questions so far? I think next we're going to talk about another type of chemical reaction, the opposite of dehydration synthesis, actually. 3:45, okay. Oh, no questions. So we talked about a dehydration synthesis, how all of these massive molecules are constructed by linking together monomers at the expense of removing water molecules, okay? Well, let's talk about the opposite situation, where instead of removing water molecules to link monomers together, what will we be doing? I'll give you a second to write it. It's the opposite in every way. What's our next chemical reaction? Adding water, totally. So instead of removing water, we're going to add water to break molecules apart, totally. So let's look at that. It's called hydrolysis. It's a hydrolysis reaction. It's the opposite of dehydration synthesis. So I'll erase this. Okay, hydrolysis. And again, the word tells us what is going on. Hydrolysis. So we're going to break it apart and break it apart. Ha. We're going to break it down and look at the word and decipher why it's called hydrolysis, and it makes sense. Let's write its definition first. This is a chemical reaction that breaks polymers into monomers. Polymers into monomers. Since water is required, I'll use blue. Hydrolysis. Okay, well let's look at lysis first. Now, lysis comes from some Greek root or something, but biology, you're gonna find this a lot this semester, is learning a new language. It's not like learning a new language. It is learning a new language. There are so many words that we use that are just totally brand new. Just like if you're learning something other than English, you learn all these new words. Lysis is one of them that biologists use a lot. For instance, viruses will lyse host cells. And to lyse a cell, or lyse something, lysis, it means to break it. To break it open or split it apart, that's a lyse, or to lyse something. So lysis is to break. So actually how viruses cause harm is they replicate, they divide, or they duplicate, they make more and more viruses within our cells until, this is one way viruses do it, they burst the cell apart and they lyse the cell. Viral lysis. It's not good, but that's how they can do stuff, or that's part of their reproductive cycle. Okay, and then hydro, right? Water. In this case, adding water, right? Add water. Okay. For hydrolysis, we're just going to do, for our example, we're just going to do the reverse of what we did for dehydration synthesis. We're going to take lactose and break it apart, this time into glucose and galactose, okay? So we're starting with lactose, our disaccharide. Starting this time with lactose. And like we said, this time we're adding water. So I'm going to write plus H2O. And we have our reaction arrow, getting crazy with the colors today. And I'm going to mention something very important, like very, very important in biology, whenever we're talking about, just because this what I'm about to say, it's a famous example. What I'm about to, lost my train of thought here. Whenever we're doing a chemical reaction within a cell, we need something called an enzyme. And an enzyme, we write on top of the reaction arrow. We have a whole lecture on enzymes, so don't worry too much about it. I just want to throw it out there though, so it's kind of implanted in your mind. I kind of plant the seed, so when we get into enzymes, have more of a relevance. But the enzyme involved, enzymes catalyze reactions. They cause them to happen in our cells, and all of them need an enzyme, by the way. So they're very important. The enzyme here is called lactase. So I'm just going to write lactase here. And this is our enzyme. And the enzyme lactase is going to catalyze this breakdown of lactose with water to form glucose and galactose again, like we had before. Oops. And there's gonna, since the water comes apart and adds its two groups again, we'll have an OH here and an H on this molecule. And that's it. And we have glucose and galactose again. Hydrolysis is, I mean, hydrolysis and dehydration synthesis are are fundamental, very important processes in the cells for building macromolecules like DNA and like polysaccharides. And hydrolysis is so important in digestion. In fact, all our digestive enzymes like lactase, this is how we break down lactose, how we get energy from it. So it's very, very useful. Okay. How are we doing on time? Oh, we're good, okay. So, any questions before we move on to our next class, next category of biological molecules? Proteins are next. We did carbohydrates. Nothing, huh? All righty, cool. So let's move on. Let's move on to proteins. Proteins. This is our second class of biological molecules that we'll be discussing. Okay. Number 2, proteins. All right, so proteins have a monomer, repeating unit to form polymers of proteins, and the monomer is called an amino acid. So the monomer is called an amino acid. Amino acid. So proteins are composed of amino acids. There are 20 different amino acids. So I'm going to write 20 different types. Different amino acids. So I remember there's a question on your lecture quiz about the difference in structure of these amino acids. And we're going to take a look at that compared to the similarities between them. Well, let's first point out the difference between each of the 20 amino acids. And I'll show you some examples on the slide as well, but I'll underline this. So the 20 different amino acids vary. They're different due to their chemical side chains. We'll take a look at this. So they vary by different types of chemical side chains. Side chains being the important thing. Let's just keep it at that. All right. Side chains, that's how they're different. Well, first, let's look at a typical amino acid on the slide here. Okay, well, here are 4 amino acids. We have phenylalanine is one, asparagine, aspartic acid, and proline. Okay? And I want you to notice that there's this, well, first, I want you to notice that there's this similar looking carbon backbone to all of the amino acids. Not these 4, all 20 have this very similar, pretty much the same, except proline's a little bit of an exception. It has this, it's attached, it's a little weird, but 19, but it's similar. Just here, take a look. You have this central carbon, right, in the middle, attached to a hydrogen for all of them, right? Again, he's a little strange, but you can see that hydrogen. And then you have what we call an amino group, which is this nitrogen attached to the hydrogens on the left side of them. And you have a carboxyl group. You don't have to know the names of these. Well, you do for the quiz, only because the quiz is going to ask you, if you get this question, it's from a bank of questions, what is different between amino acids? And it's these side chains, it's these highlighted regions that differ between the 20 amino acids. So these are regions that stick off this central carbon, and you see they're highlighted and they look completely different, right? And it's very important. It actually gives the amino acids different properties. As you can see, this one's a non-polar amino acid. This one's polar. This one's ionizable, so it has a charge to it. And that all is going to actually affect how the protein bonds. It can form an ionic bond if it has this charge versus a hydrogen bond with the polar group. So that's going to actually affect the structure of the protein. What kind of bonds are involved might affect the shape. And that's very important. Like I said, biology is this interconnection between shape and structure. They're one and the same. But for our purposes, I just want you to know that the difference is those side chains. Sometimes, I think on the quiz, oh no, maybe I added side chains. R groups, they're sometimes called R groups because to be lazy, not lazy, but to shorten it, sometimes instead of the whole group written off attached to the carbon, they just write an R. So sometimes R group, side chain, same thing. So that's the difference between amino acids. And there's 20 of them. So those are the monomers, right? What happens is, do a different color here, is again, just like we saw with carbohydrates, we're gonna be linking them together and breaking them apart. So if I have amino acids here that I'm gonna represent as circles, and I often abbreviate amino acid as AA. So if I have 3 amino acids linked together here, and I want to add another one on. Oh, we have a question? Question. Correct, Sophia, that's correct. The difference between amino acids are their R groups. They're all different from one another. Exactly. Yeah, that's like, if you know that, you're walking into major biology, biochemistry with, you know, a head start. That's just knowing that basic amino acid structure and the difference. That's huge. Okay. Amino acids, and if I'm going to add another amino acid onto this chain, what kind of reaction would that be? What kind of chemical reaction if I wanted to link another amino acid to this chain of amino acids here? Right, dehydration synthesis. So I'd have an OH and an H. They would pop off, and water would come off and form as a byproduct, and we link together this additional amino acid, exactly. And if I wanted to do the reverse reaction, go this way, and I wanted to break off one of these amino acids with the addition of water, right? Just do the reverse here and break off this amino acid. What reaction is that? Hydrolysis. Cool. Awesome. All right. Okay, perfect. So, ow, those were my knees. Oops. Okay. So this might be the well, we'll see how far we get. Let's name a few proteins and why they're so crucial. I know I say everything's important, but if I had to choose a biological molecule, if I had to choose the most important one, I'd probably choose proteins. Carbohydrates, lipids, and nucleic acids are really important. But proteins, you're about to see, kind of allows us to exist. It allows us to, even for me to move my hands, and you're going to see why. So we're going to look at a few examples of proteins and their function. It's not a universal function. Proteins have a variety of different functions, depending on what protein we're talking about. Okay, so we'll have examples, polymers of these amino acids together, strung together, and their specific function. The fancy term for a polymer, a protein polymer, is polypeptide chain. But yeah, I might as well write it. Let's write some examples of a polypeptide chain, of polypeptide chains. So the polymer is called polypeptide chain of amino acids, right? Let's do examples and functions. First one, keratin is a protein that we find in hair, skin cells, and nails. Keratin is what it's called. And its function is support, structural support. You know, our skin cells are just jam-packed with keratin. Not just structural support, but also protection. It provides a barrier, keratin. So I'll add that in as well. Okay. Myosin and actin are two proteins that are they actually make up muscles. So myosin and actin in muscles, the function of which would be movement. So muscles are made of proteins, these polypeptide chains. I mentioned with lactose being broken down by this enzyme. Enzymes are also proteins. We have if you take any biochemistry and learn it, and we're gonna do a little slice of it in metabolism, we're gonna look at a lot of chemical reactions in cells. Each one has an enzyme that'll allow it to proceed, allow the reaction to happen. So enzymes are catalysts of chemical reactions. So lactase, what we looked at earlier, that allowed the breakdown and promoted that reaction to go. It's a protein. Lactase is a protein, it's an enzyme. A lot of important chemical reactions. Another good example of a protein is hemoglobin. Anyone know what hemoglobin does? Where we find it, what it does? It's involved in the transport of something. It's in the blood, totally. Red blood cells, what is it doing? Why is it in our red blood cells? Yeah, transports oxygen, exactly, Anastasia and Sophia. Yeah, so transports oxygen. In red blood cells, you can add that if you'd like. In this amazing, there's so many incredible functions for proteins that I'm running out of room. And there's another incredibly important one. Antibodies are proteins. Antibodies are involved in our immune system to fight bacteria, viruses, pathogenic microorganisms that can cause disease. So let's write defense. / fighting infection. No problem, Elizabeth. So talk about exactly. So enzymes are going to be a large focus for our next unit, actually, which is going to be when we talk. We do an entire lecture on enzymes and how they catalyze chemical reactions, what they're doing specifically, and how they're causing them to proceed and happen. And then we're also going to talk in metabolism. When we do cellular respiration, it's a lot of chemical reactions going on in the breakdown of food, and each reaction requires an enzyme. So we'll be looking at a lot of enzymes. Yeah, they enable breakdown, and in fact, there is also an enzyme required for every dehydration synthesis reaction. There's an enzyme required for building molecules as well. All chemical reactions in cells. And for the immune system, yes, absolutely. So antibodies are produced by cells of the immune system. Correct. Do we have time? No, we're out of time. It's okay. This is where I ended with my other school too. So next time we'll be discussing, still with proteins, they're a little more on their structure and the different levels of protein structure. We'll start here next time. I'm thinking we take a 10-minute break before we do the lab mini lecture. How does that sound good? So meet back here at 4:25. Coolio? All right. 4:25, meet back here, and we'll talk about this week's lab on biological molecules.